It's really amazing when you think about it. Like, you start as just one single cell, and then boom,
Yeah.
all these intricate steps happen, and you become this, complex being
Yeah.
with trillions of cells, all specialized, all working together.
It's incredible, isn't it?
It really is. And what I find really fascinating is that, like, in those very early stages, if you were to look at a human embryo next to a chick embryo, like they show in figure 47.1,
Right.
it's pretty striking how similar they look.
Yeah, they're remarkably similar.
Really makes you wonder about, like, the basic tools that nature uses to kind of build life.
Yeah, absolutely. It does, like you said, that similarity across such different creatures, it really points to, a very conserved set of processes at work during development.
Right.
And that's exactly what we're going to be looking at in this deep dive.
Yeah.
Unpacking those essential mechanisms that take, what seems like a simple egg and turn it into a structured embryo. So we'll be using the material that we have to kind of uncover the key theories, concepts, important research findings, and even some case studies that really highlight, this remarkable transformation.
That's awesome. And to really kind of understand these universal principles, I mean, scientists, they often use these things called model organisms, and they're really essential tools in developmental biology.
Oh, yeah, for sure.
And we're talking about things like sea urchins and fruit flies and frogs and chicks and even like the little nematode worm.
Right.
And, each one of these has its own unique advantages for studying specific aspects of development.
Exactly.
Like the sea urchin that we talked about in the material is a great example.
Mm.
It releases its eggs and sperm right into the water. So it makes it so easy to study those early cell interactions during fertilization.
Yeah, it's really accessible for lab study for that reason.
Yeah, exactly.
Hmm.
Okay, so why don't we just jump right in and start at the very beginning with fertilization and cleavage.
Okay, so fertilization is really the foundational event. It's the fusion of a haploid egg and a haploid sperm, each carrying half the genetic information,
Right.
to create a diploid zygote, which now has the full set.
Okay.
But it's more than just combining DNA. It's this union that really kicks off the whole developmental program.
Yeah, it triggers everything.
Exactly. In sea urchins, this is a very carefully orchestrated process, starting with the sperm being attracted to the egg by these chemical signals that are released from the egg's outer layers.
Oh, so the egg's kind of like calling out to the sperm.
Yeah.
Interesting. And then you have this thing called the acrosomal reaction.
Yes.
And I mean, figure 7.2 shows it so well. It's like the sperm has this little compartment at its tip
Mm-hmm.
called the acrosome, and it's packed with these hydrolytic enzymes.
Powerful enzymes.
Yeah, so what do these enzymes do?
So these enzymes are released, and basically they digest a path through the egg's jelly coat, that protective outer layer.
Right.
And this allows something called the acrosomal process, which is like a little projection that extends from the sperm to reach and bind to the egg's plasma membrane.
Okay, so it's kind of like clearing a path and then reaching out.
Exactly. And it's not just any binding. It's a very specific lock and key kind of recognition
Oh, wow.
between certain proteins on the sperm and matching receptors on the egg.
So they have to fit together just right.
They do, and the specificity is super important, especially in species with external fertilization, because it makes sure that a sperm only fertilizes an egg of the same species.
Right, that makes sense.
Yeah.
So once they've kind of, made that connection, what happens next?
So this species-specific binding triggers the fusion of the sperm and egg plasma membranes.
Okay.
And as soon as that happens, ion channels in the egg's membrane, they open up, and this leads to a really quick influx of sodium ions.
I see.
This causes a change in the electrical potential across the membrane called depolarization.
Okay.
And that acts as a quick defense against something called polyspermy.
Polyspermy, what is that?
It's when an egg is fertilized by more than one sperm.
Oh, so like multiple sperm getting in.
Exactly. And as you can imagine, having extra chromosomes would be a really bad thing for the developing embryo.
Right. It would mess everything up.
Yeah, so this fast block to polyspermy, it kicks in within seconds of the sperm binding.
Wow. That's fast. So it's like a really quick first line of defense?
Exactly.
But that's not the only way that the egg prevents polyspermy, right?
No, you're right. There's also a slower but more permanent defense mechanism called the cortical reaction.
Right. I remember seeing that in figure 47.2 too.
Yeah, and it involves these small vesicles inside the egg's cytoplasm called cortical granules.
Okay.
So when the sperm binds these granules, fuse with the egg's plasma membrane and release their contents into the space between the plasma membrane and the vitelline layer.
The vitelline layer, remind me what that is again.
Oh, it's like a fibrous mat that surrounds the egg.
Right, okay.
So the release of these cortical granule contents, it sets off this whole chain reaction.
Like a domino effect?
Yeah, exactly. So enzymes, they clip off those sperm binding receptors that we talked about.
Oh, wow. So no more sperm can bind.
Exactly. And importantly, the vitelline layer, it lifts away from the egg surface and hardens into this tough fertilization envelope.
Oh, so it's like an extra barrier.
It is, and it acts as a slow block to polyspermy,
Okay.
providing a long-lasting barrier to make absolutely sure that only one sperm's genetic material gets in.
So it's like a double security system.
Yeah, you could think of it like that. And this dual system really emphasizes how important it is to prevent multiple sperm from fertilizing an egg.
Right. It's like essential for proper development.
It is across the entire animal kingdom.
Wow. And this whole cortical reaction is triggered by a wave of calcium ions, right?
Yes, that's right.
And in figure 47.3, they showed this really cool experiment where they used this fluorescent dye that lights up when it binds to calcium.
Oh, yeah.
And they actually saw this wave of calcium sweeping across the egg, starting from where the sperm entered.
That's pretty amazing.
It is, and it happened right as the fertilization envelope was forming.
It's a beautiful demonstration of how these simple ionic signals can start such big developmental changes.
Yeah, and it's not just the cortical reaction that this calcium surge triggers, right? It also activates the egg.
You got it.
So what does that actually mean? Like, what happens?
Well, the egg's metabolism just goes into overdrive.
Okay.
Cellular respiration increases like crazy, and new protein synthesis ramps up.
So it's like the egg wakes up and is ready to go.
Exactly. It's preparing for this huge task of building an embryo. And what's really interesting is that scientists have found that you can actually artificially activate an egg just by injecting calcium.
Really?
Yeah, even if you remove the nucleus.
Wow, so the egg has everything it needs already.
Exactly. All the cellular machinery for activation is already there in the cytoplasm. It's just waiting for that calcium signal.
That's amazing. And it's really interesting how many things about fertilization are the same across different species.
Right.
But our material also mentions some key differences, like with sea urchins. The eggs are released after they've finished meiosis. You know that process that halves the chromosome number? But in humans, the eggs are arrested at a specific stage of meiosis called metaphase 2.
Yes.
And they only finish meiosis after a sperm enters.
That's right. And it's a really important difference. It highlights that the timing of these cellular events can vary quite a bit.
Right. And when you look at mammalian fertilization in figure 47.4,
Yes.
there are even more differences.
You're right. Like, internal fertilization, for one, means that the sperm has to travel a lot further.
Yeah, get a longer journey.
Yeah, first it has to navigate through the follicle cells surrounding the egg, and then it has to penetrate the zona pellucida, which is another layer of extracellular matrix.
Right.
And in mammals, the acrosomal reaction is actually triggered by the sperm binding to specific receptors in the zona pellucida.
Oh, so it's different from the sea urchin.
Yeah, and while mammals do have a cortical reaction that modifies the zona pellucida,
Okay.
which acts as a slow block to polyspermy,
Mm-hmm.
we haven't found a fast block like the membrane depolarization we see in sea urchins.
Interesting. And the whole process in mammals is a lot slower, right?
It is, yeah. The first cell division can take anywhere from 12 to 36 hours.
Wow. That's a big difference compared to sea urchins.
Yeah, which is pretty quick, about 90 minutes.
Yeah. Okay, so after fertilization, we move on to the next big stage, which is cleavage.
Right.
And figure 47.5 gives us a good overview of how these early cell divisions are different from typical cell division.
Mm-hmm.
So instead of the embryo getting bigger overall, it's a series of really rapid mitotic divisions
Right.
that divide that big cytoplasm of the zygote into a bunch of smaller cells called blastomeres.
Exactly. It's kind of like taking a big cake and cutting it into smaller and smaller pieces.
Yeah, I like that analogy. And what's the point of doing that?
Well, the key here is that these rapid divisions help to restore the balance between the cell's nucleus and its cytoplasm.
Okay.
You see, early development is mostly fueled by the mRNA and proteins that were already packed into the egg when it was formed in the mother's body.
Right, during nucleogenesis.
Exactly. So cleavage ensures that each of these new, smaller blastomeres has a nucleus and a manageable amount of cytoplasm to work with as development continues.
So it's kind of like giving each cell its own little starter kit.
Yeah, a good way to put it.
And after a bunch of these divisions, like 5 to 7 rounds,
Mm-hmm.
the embryo turns into a blastula,
Yes.
which is that hollow ball of cells with a fluid-filled cavity inside called the blastocoel.
You got it.
And these cleavage divisions, they don't happen the same way in all animals, right?
No, they don't.
Like figure 47.6 shows that uniform cleavage in sea urchins, where the divisions are very symmetrical.
Right.
But then if you look at frogs in figure 47.7, it's different.
Yeah, it is.
And that's because of the yolk, right?
Yes, the yolk is unevenly distributed in the frog egg. It's concentrated at the vegetal pole, which is the bottom part of the egg,
Okay.
and this big clump of yolk makes it harder for the cleavage furrow to cut through that area.
So it's like an obstacle course for the cleavage furrow.
Kind of, so you end up with smaller blastomeres in the animal hemisphere.
That's the top part.
Right, and the blasticle gets pushed up towards that animal pole.
Okay, so it's all squished up there.
Yeah, you could say that.
And in frogs, the cleavage furrow still manages to divide the egg completely.
Mm-hmm.
So that's called holoblastic cleavage, right?
Yes, meaning complete division. And this type of cleavage also happens in other animals that don't have a lot of yolk,
Like what kind of animals?
like echinoderms, mammals, and annelids.
Okay,
And in those cases, the blasticle usually forms more in the center of the blastula,
right.
and the blastomeres are more similar in size, at least in the beginning.
But then you have animals with eggs that are packed with yolk.
Yes.
Like birds, reptiles,
Right.
lots of fish, and insects.
Right.
And in those cases, there's so much yolk that the cleavage furrow just can't get through it all the way.
Exactly, and that's called meroblastic cleavage.
Partial cleavage.
Yeah, so in chicks, for example, cell division is limited to this small yolk-free disc of cytoplasm up at the animal pole.
Yeah.
And this forms a cap of cells called the blastodisc.
So it's like a little island of dividing cells on top of the yolk.
Yeah, that's a good way to visualize it.
And we can't forget about Drosophila and other insects.
Right.
They do this really interesting thing called superficial cleavage.
Yeah, it's pretty unique. So at first, the zygotic nucleus divides multiple times, but without any cytokinesis.
Meaning the cells don't actually separate.
Exactly. So you end up with this one big cell with hundreds of nuclei scattered throughout the yolk.
Wow.
And then these nuclei, they migrate to the edges of the cell, and only then does the plasma membrane start to fold in between them.
So it's like they form a blastula from the inside out.
Kind of, yeah. It's amazing how many different ways this initial partitioning of the zygote can happen.
It really is. And what decides when this rapid cleavage phase ends?
Right.
Like what tells the embryo to move on to the next stage of development?
Well, our material mentions this scientific skills exercise where they looked at frog embryos.
Okay, what did they find?
They found that during cleavage in frog embryos, the cell cycle is super short. It's mostly just the S phase, which is DNA synthesis, and the M phase, which is mitosis, with really short or even no gap phases, G1 and G2.
Okay.
But then around the 12th cell division, the G1 and G2 phases start to get longer.
Oh, so they're not just dividing like crazy anymore.
Exactly, and the cells actually start to grow.
Okay.
They start making new proteins and organelles.
So they're becoming more than just copies of each other.
Right, and at first they thought that maybe reaching a specific ratio of nucleus to cytoplasm might be the trigger.
Okay.
But later experiments showed that it might be more about the number of cell divisions.
Interesting. So like a built-in timer?
Yeah, it seems like there's some kind of developmental clock that controls how long the cleavage phase lasts.
Okay, so after all this rapid cell division, without much growth, the embryo needs to start taking shape.
Right.
And that brings us to morphogenesis, which is described in section 47.2 as the processes that actually shape the animal body.
Yeah, and this crucial phase includes both gastrulation and organogenesis.
Okay, so let's start with gastrulation.
All right, so gastrulation is basically this big reorganization of the blastula into a multi-layered structure called the gastrula.
Okay.
It involves lots of carefully coordinated cell movements where cells change their positions and make new connections with their neighbors.
So it's like a big cellular dance.
Yeah, that's a good way to think about it. And this process is really important because it creates the 3 primary embryonic germ layers.
Okay, remind me what those are again.
Sure, so you have the ectoderm, which forms the outer layer of the embryo and eventually gives rise to the skin and nervous system.
Right.
Then you have the endoderm, which lines the developing digestive tract and other internal organs.
Okay.
And in triploblastic animals,
Which includes us.
yes, you have the mesoderm,
Okay.
which forms between the ectoderm and the endoderm, and it will give rise to muscles, bones, blood, and lots of other tissues.
Wow, so these are the building blocks for all the different parts of the body.
They are, and figure 47.8 gives you a really good visual of these fundamental changes.
Yeah, it's really helpful. Okay, so how does this gastrulation process actually get started?
Well, it often begins with a process called invagination.
Invagination, what's that?
It's where a sheet of cells folds inward or buckles,
Okay.
creating this pouch-like structure called the archenteron.
Which is the primitive gut.
Right, and an opening called the blastopore.
Okay.
And it's interesting to note that in diploblastic animals like jellyfish, which only have two germ layers, the ectoderm and the endoderm, the blastopore is the only opening to their gastrovascular cavity.
So it acts as both the mouth and the anus.
Exactly, but in triploblastic animals, it's different.
Right, because we have that third germ layer, the mesoderm.
Exactly. In protostomes, which are a big group of animals that includes things like mollusks and insects, the mouth develops from the blastopore.
Okay.
But in deuterostomes, which includes us and other vertebrates, the anus develops from the blastopore.
Oh, wow.
And the mouth forms somewhere else.
That's a pretty big difference.
It is, and it's a key distinction in how animals evolved.
Okay, so let's look at how gastrulation happens in some of the model organisms we've been talking about.
Okay, so in frogs, figure 47.10 shows how it starts on the dorsal side of the blastula with an invagination forming the blastopore and its dorsal lip.
Okay.
Then cells from the animal hemisphere roll inward over this dorsal lip in a process called involution.
Involution.
Yeah, and they eventually form the endoderm and mesoderm.
So it's like they're tucking themselves in.
Right, and at the same time, the cells that stay at the animal pole spread out to cover the whole embryo, becoming the ectoderm.
So they're kind of like stretching out.
Yeah, and as all this is happening, the blastocod, that fluid-filled cavity, shrinks because the argenteron, the developing gut, is expanding.
Oh, so it's like one is taking over the space of the other.
Exactly, and then towards the end of gastrulation in frogs, you even see a plug of yolky cells called the yolk plug sitting in the blastopore.
Okay, and in chicks with their yolk-rich eggs and meroblastic cleavage, gastrulation looks pretty different, right?
It does, yeah. Figure 47.11 shows that. So remember they have the epiblast and hypoblast layers?
Right.
And all the cells that will make up the actual embryo, they come from the epiblast.
Okay.
During gastrulation, some of these epiblast cells migrate towards the middle of the blastodisc.
Okay.
Then they detach and move inward, forming this thick band of tissue called the primitive streak.
Primitive streak.
Yeah, some of these cells move down and displace the hypoblast, forming the endoderm,
Okay.
while others migrate to the sides and form the mesoderm.
Okay.
And the remaining epiblast becomes the ectoderm.
So it's like a very organized migration.
It is, and it's important to note that the primitive streak in chicks is basically the same as the blastopore lip in other vertebrates.
Okay.
It acts as the site where cells move inward.
And even in humans, with our relatively small yolk-pore eggs, gastrulation still involves the primitive streak.
You got it. Figure 47.12 shows that.
Yeah.
So after the blastocyst,
The mammalian blastula.
right after it implants in the uterine wall, the inner cell mass differentiates into the epiblast and hypoblast. And just like in chicks, the embryo mainly develops from the epiblast cells.
Okay.
And gastrulation involves those epiblast cells moving inward through the primitive streak to form the mesoderm and the endoderm.
So even though the blastula looks different at the beginning because of the amount of yolk,
Right.
the basic process of forming those germ layers through cell migration and invagination is pretty much the same.
It is a very conserved process.
And what's also really cool is that amniotes, which are reptiles, birds, and mammals, they develop these extra embryonic membranes.
Yes.
The chorion, amnion lantua, and yolk sac.
Right, as shown in figure 47.13.
Yeah, these are like really important adaptations that allowed development to happen on land.
Absolutely. They provide the self-contained life support system either within the egg or the uterus.
So it's like the embryo has its own little world.
Yeah.
A good way to put it.
So can you explain what each of those membranes does?
Sure. So the amnion creates this protective fluid-filled sac around the embryo,
Okay.
which keeps it from drying out and cushions it.
Like a little waterbed. Yeah. And the chorion is the outermost membrane, and it's responsible for gas exchange.
So it's like the embryo's lungs.
Exactly. And the allantois helps get rid of waste. And in mammals, it also contributes to the blood vessels in the umbilical cord.
Oh, wow.
And the yolk sac, well, in reptiles and birds, it encloses that nutrient-rich yolk.
Right.
But in mammals, it's an early site for blood cell formation.
Okay, so they all have slightly different roles.
They do, but they're all essential for development on land.
Yeah. The evolution of these membranes was a big step for vertebrates.
It was, for sure.
Okay, so once gastrulation has set up those germ layers,
Right.
the next step is organogenesis, the process of actually forming organs.
Yes.
And in section 47.2, it says that this involves some really complex interactions between cells from different germ layers.
It does.
And a classic example is neurulation in vertebrates, which is the early stage of forming the brain and spinal cord.
Right. And figure 47.14 goes through that process in detail.
Yeah, it's really helpful to see it visually.
Yeah.
So how does neurulation begin?
It starts with the formation of the notochord.
The notochord.
Yeah, it's this flexible rod of mesodermal cells that runs along the back of the embryo.
Okay.
And this notochord acts as a signaling center.
Yeah.
It releases molecules that tell the ectoderm above it to thicken and form the neural plate.
So it's like the notochord is giving instructions.
Exactly. It's a prime example of inductive signaling.
Which is where one group of cells directly influences how another group develops.
Right. And then the neural plate goes through some dramatic changes in shape.
Like what kind of changes?
It folds inward and eventually pinches off to form the neural tube.
Which will become the brain and spinal cord.
You got it.
And as this neural tube is forming, another really interesting group of cells appears at the edges of the neural plate.
Yes, the neural crest cells.
Hey, what's so special about those?
Well, they migrate all over the developing embryo.
Really?
Yeah, and they contribute to a ton of different tissues and structures,
Like what?
like peripheral nerves, pigment cells, parts of our teeth, even some bones and cartilage in the skull.
Wow, so they're really versatile.
They're a great example of how flexible early embryonic cells can be.
That's amazing. And along with neurulation, we also see the formation of somites,
Right.
which are those blocks of mesodermal tissue that form in pairs along the notochord.
Yes, and they're really important for setting up the segmented structure of the vertebrate body.
So that's where our vertebrae and ribs come from.
Exactly. They also give rise to the muscles of the back and limbs.
It's really amazing how these early events lay the foundation for the whole body plan.
It is. It's incredible.
And it's interesting that our material compares neurulation in vertebrates, which happens on the back of the embryo,
Right.
to neurulation in insects, which happens on the belly side.
Yes.
But even with this difference in location,
Mm-hmm.
the basic process of a sheet of ectodermal cells rolling up into a tube and the molecular signals involved are very similar.
That's right. It suggests a deep evolutionary connection.
Yeah, like a common ancestor way back when.
Exactly. And it shows how effective these developmental mechanisms are.
Right. And morphogenesis isn't just about forming these big organ systems.
No, it's not.
It's also about really precise changes in cell shape and position at a much smaller scale.
Yes, and our material talks about how the cytoskeleton plays a huge role in these cellular transformations.
The cytoskeleton, remind me what that is again?
It's that internal network of protein filaments, like microtubules and microfilaments.
Right.
So, for example, during neural tube formation, microtubules help to lengthen the cells of the neural plate,
Okay.
while actin filaments contract at the top of the cells, making them wedge-shaped.
Okay.
And that leads to the bending and folding of the tissue.
So it's like the cells are being molded into the right shape.
Exactly.
And then there's this really cool process called convergent extension,
yes.
which is shown in figure 47.17, where a sheet of cells gets narrower and longer.
Right.
And this happens when individual cells stretch out and then squeeze in between their neighbors.
Yeah, like a crowd of people trying to get through a narrow doorway.
Yeah, and this type of movement is super important in lots of developmental events.
It is like lengthening the primitive streak during gastrulation,
Okay.
and making the archenteron longer.
Wow. And of course, cell migration is another key part of morphogenesis.
Absolutely. We already talked about how neural crest cells migrate,
Right.
but tons of other cell populations also move in a very controlled way to reach their final spots in the embryo.
And how do they know where to go?
Well, this movement often relies on amoeboid-like motion, which is driven by changes in the cytoskeleton,
Okay.
and also on interactions between cell adhesion molecules on the cell surface and the extracellular matrix.
The ECM.
Yes, which is that network of proteins and carbohydrates that provide support to cells.
Right.
And scientists are even looking at ways to use artificial ECMs to guide cell migration
Well,
for things like tissue repair and regeneration in medicine.
that's really cool. So they're kind of hijacking these natural processes.
Yeah, in a way.
Okay, and lastly, in this section on morphogenesis, the material talks about programmed cell death or apoptosis.
Right.
And it's interesting because it's not just about building things up.
It's not.
It's also about getting rid of cells and tissues at specific times and places during development.
Exactly. A classic example is when a tadpole loses its tail during metamorphosis.
Right.
But apoptosis also plays a really important role in other things, like making sure there's the right number of neurons in the nervous system,
Okay.
getting rid of immune cells that might attack the body's own tissues,
Oh, wow.
and even shaping structures like our fingers and toes by removing the webbing between them.
Yeah, they show that in figure 11.21.
Right. It's a crucial process for making sure the organism ends up with the right final form.
So it's kind of like sculpting the body.
Exactly.
Okay, so now let's shift gears a bit and talk about how cells actually get their specific identities and roles during development.
All right.
So section 47.3 dives into how cytoplasmic determinants and inductive signals control cell fate. And it starts by defining some key terms,
Right.
like cell fate,
Which is basically what a cell will become.
like its final job in the body.
Exactly. It's structure, function, and characteristics.
Okay.
Then there's determination, which is the process where a cell commits to a certain fate.
Even though it might not look like that cell type yet.
Right. And then differentiation is when the cell actually goes through the changes in gene expression and morphology to become that specialized cell.
Okay, so it's like choosing a major in college.
Yeah.
That's determination.
Right.
And then actually taking the courses for that major is differentiation.
I like that analogy.
And the big idea behind all this specialization is differential gene expression, right?
Absolutely.
Every cell in an animal has the same set of genes,
The genome.
but they become different by expressing different subsets of those genes.
Right. It's kind of mind-blowing when you think about it.
It is, and it's amazing that even across very different animal body plans, a lot of the basic developmental mechanisms and the genes that control them are very similar.
Yeah, they're highly conserved.
Like the PAX6 gene,
Yes.
which controls eye development in both vertebrates and Drosophila.
It does.
And they even showed that the mouse version of that gene can make an eye form in a fly.
It's a really powerful example of how deeply conserved these developmental mechanisms are.
So how do scientists figure out how these differences in gene expression happen and how cells decide what they're going to be?
Well, they use something called fate mapping.
Fate mapping.
Yeah, it's basically tracing the lineage of cells back to their origins in the early embryo.
Oh.
And our material mentions how early fate maps were made just by watching what happened.
Okay.
Like Walter Voigt's experiments with frog blastulas.
Right. Figure 47.18a.
Yes. He used vital dyes to mark specific groups of cells at the blastula stage.
Okay.
And then he could see where those marked cells ended up in the later gastrula.
Oh, that's clever.
It is. And later on, scientists developed more sophisticated techniques to mark individual cells and track all their descendants.
Like in the cell lineage analysis of the tunicate in figure 47.8b.
Exactly.
And then there's that incredible work that's been done with the nematode worm C. elegans,
Oh, yeah.
as shown in figure 47.19.
It's amazing work.
It is, because this worm is transparent and it always has the same number of somatic cells.
905.9 in the adult hermaphrodite.
Scientists were able to map the entire developmental history of every single cell.
It's pretty mind-blowing.
It is, they could literally track every cell division from the zygote to the adult worm.
It's been a really valuable resource for understanding how development works.
Yeah, that's incredible. And that super detailed cell lineage map of C. elegans has given us so much insight.
That has.
Like, for example, it's helped us understand how germ cells become determined.
Those are the cells that will eventually make sperm and eggs.
Right, and in C. elegans, these cells have these things called P granules.
Yeah, which are these dense clumps of RNA and protein. And figure 47.21 shows how these P granules are spread out in the fertilized egg at first.
Okay.
But then as the cells divide, they get split up unevenly.
Okay.
So they end up only in the cells that will become the germline.
So it's like the P granules are determining the fate of those cells,
They act as cytoplasmic determinants.
which are factors that are present in the cytoplasm of the egg.
Right.
And because they're not evenly distributed, they can influence what the daughter cells become.
Exactly.
And C. elegans has also been super important for figuring out how programmed cell death works, right?
Yes, definitely. The cell lineage analysis showed that exactly 131 cells undergo apoptosis during normal development.
Apoptosis is another word for programmed cell death.